The discovery of DNA. What makes us who we are? What instructs a seed to become a tree, a zygote to become a human, or a single fertilized egg to create trillions of cells, working in harmony? The answer lies deep within the nucleus of every cell, in a twisting, microscopic thread of life, DNA. But this knowledge, so vital to our understanding of biology and medicine, wasn't always known. It took decades of work, failures, and groundbreaking insight to uncover. Join us on a journey, through time, ambition, controversy, and brilliance, to discover why is the man who laid the very foundation of genetics, who unlocked the first secret of heredity, still considered one of science's unluckiest figures? Why did the woman whose work provided the single most critical clue in one of history's greatest discoveries die in obscurity, her name almost lost to the footnotes of a Nobel Prize she was denied? And why is it that a respected botanist, trusted by the scientific community, would unknowingly bury the truth for decades? And how we discovered DNA, the molecule of life. The story of DNA is more than a story of a molecule. It is a human epic, spanning 2 centuries. It's a tale of quiet genius, of bitter rivalries, of ambition, injustice, and a relentless quest to answer the most profound question of all. What is the code that writes the symphony of life? The year is 1822, in the small village of Heinzendorf, in the Austrian Empire. Gregor Mendel is born into a family of poor farmers. From childhood, he is curious, questioning, and deeply observant. Mendel's father teaches him the value of hard work, but poverty shadows every step of his early education. He excels in school, but his family cannot afford to support his higher studies. In his early 20s, Mendel joins the Augustinian monastery in Brno, a decision that will shape the rest of his life. The monastery gives him access to books, ideas, and most importantly, a garden. In the monastery garden, Mendel begins his now legendary pea plant experiments. For 8 years, he crossbreeds varieties of peas, carefully recording every trait: flower color, seed shape, plant height. Mendel's meticulous mind saw something different. For nearly a decade, he cultivated and tested almost 30,000 pea plants. He observed their height, their seed color, their flower shape. And he counted. He notices patterns. Some traits disappear in one generation, only to reappear in the next. Ratios emerge, not random, but mathematical. He didn't see blending. He saw patterns. He saw that traits were passed down in discrete, predictable units. A tall plant and a short plant didn't make a medium plant. They made tall plants, and only later would the short trait reappear, intact. He called these units factors. Today, we call them genes. He had written the fundamental laws of heredity. In 1865, he published his work, Experiments in Plant Hybridization. It landed with a deafening silence. The world wasn't ready. To gain recognition, he sent his paper to the most eminent botanist of the era, Carl Nageli. Carl Wilhelm von Nageli, the respected botanist who missed history. Born in 1817 in Switzerland, Nageli is celebrated for his work in plant anatomy and cell theory. He is meticulous, brilliant, and widely respected. His career is a string of successes, professorships, publications, and recognition. When Mendel contacts him, Nageli sees the experiments as interesting but flawed. In fairness, at the time, many scientists believed traits blended like paints, not segregated into discrete units. The missed opportunity. Nageli's suggestion to study hawkweed, a plant that reproduces through apomixis, leads Mendel into results that seem inconsistent with his earlier findings. Without realizing it, Nageli has steered Mendel into a dead end. Historians later debate, was this deliberate dismissal or simply the result of scientific blind spots? Whatever the case, Nageli's failure to recognize the importance of Mendel's work delayed the acceptance of genetics for more than 30 years. Mendel's elegant ratios vanished in the face of this biological complexity. He tried for years, his confidence eroding. Nageli neither promotes nor publishes Mendel's work widely. The laws of inheritance are left to gather dust in obscure journals. Defeated, he eventually abandoned his work on heredity and became the monastery's abbot, his groundbreaking discovery locked away in an obscure journal. This is the tragedy of Mendel. He was a man 35 years ahead of his time, rejected by the very authority he sought for validation. He would die never knowing the revolution he had started. On January 6, 1884, Mendel dies from kidney disease, largely unknown to the scientific world. He had no idea that decades later, his work would be called the foundation of modern genetics. The birth of a molecule, 1869 to 1920. At the exact same time Mendel was wrestling with hawkweed, in 1869, another quiet pioneer was making a completely unrelated discovery. A young Swiss physician, Friedrich Miescher, was interested in the chemistry of cells. His subject matter was not pleasant. He obtained used, pus-filled bandages from a nearby surgical clinic. In the nuclei of these white blood cells, he isolated a substance unlike anything seen before. It was rich in phosphorus and acidic. He called it nuclein. He had no idea what it was for. He speculated it might be involved in storing proteins, but its true purpose was a complete mystery. The 19th century closed with the two most important pieces of the puzzle on the table, but no one on Earth knew they were part of the same picture. The rules of inheritance had been written, and the molecule of inheritance had been found. They just existed in two different worlds. It was the first time the substance we now call DNA had been seen, but no one knew its purpose. For decades, it was thought to be just a structural support for proteins, the supposed stars of heredity. In the early 1900s, Russian-born biochemist Phoebus Levine identifies the components of nucleic acids, sugars, phosphates, and bases. But he proposes the tetranucleotide theory, which incorrectly suggested DNA was too simple to carry genetic information. The transforming principle. The year is 1900. Three different scientists, Hugo de Vries, Carl Correns, and Erich von Schermach, independently rediscover Mendel's work. Suddenly, his laws are the talk of biology. The field of genetics is born. But the central question remained, what, physically, were these genes made of? Where in the cell was this information stored? The scientific community had a clear favorite: protein. Proteins were complex, built from 20 different amino acids. They seemed perfectly suited to carry the vast complexity of life's instructions. DNA, Miescher's nuclein, seemed far too simple. A boring, repetitive molecule made of only 4 basic units, or bases: adenine, guanine, cytosine, and thymine. It was seen as mere structural scaffolding. The first major crack in the protein theory came from British bacteriologist Frederick Griffith. He was studying 2 strains of pneumococcus bacteria, a smooth S strain, which was deadly, and a rough R strain, which was harmless. 1. Inject S strain, mouse dies. 2. Inject R strain, mouse lives. 3. Inject heat-killed S strain, mouse lives. But then came the masterstroke. Griffith mixed the harmless R strain with a dead S strain and injected it. Logically, the mouse should have lived. The mouse died. And in its blood, Griffith found living, deadly S strain bacteria. Something from the dead S strain had passed to the living R strain, transforming it. He called this mysterious substance the transforming principle. He didn't know if it was protein or DNA, but he knew it was real. For over a decade, the identity of this principle remained a mystery. Then, in 1944, a team led by Oswald Avery at Rockefeller University decided to settle it. In a painstakingly systematic experiment, they took the heat-killed S strain and treated it with different enzymes. They used an enzyme that destroyed protein. The R strain still transformed. They destroyed lipids and carbohydrates. The transformation continued. But when they used an enzyme that specifically destroyed one thing, DNA, the transformation stopped. The conclusion was revolutionary. DNA was the transforming principle. DNA carried the genetic information. But the scientific world, so convinced of the protein theory, remained largely unconvinced. The proof needed to be irrefutable. That final, elegant proof came in 1952 from Alfred Hershey and Martha Chase. They used bacteriophages, Viruses that infect bacteria. A phage is little more than a protein coat surrounding a DNA core. Hershey-Chase experiment. 1. Phages are grown with radioactive sulfur, which labels the protein coat. They infect bacteria. 2. The mixture is put in a blender to knock the phages off the bacteria. 3. The radioactivity is found outside the bacteria, in the liquid. In one batch, they tagged the protein coat with radioactive sulfur. After infection, they put the mixture in a kitchen blender. The radioactivity was found outside the bacteria. The protein hadn't entered. 1. Phages are grown with radioactive phosphorus, which labels the DNA core. They infect bacteria. 2. The mixture is blended. 3. The radioactivity is found inside the bacteria. In a second batch, they tagged the DNA core with radioactive phosphorus. This time, after blending, the radioactivity was found inside the bacteria. It was conclusive. DNA, and DNA alone, was the stuff of life. The question was no longer what the genetic molecule was. The question was now, what did it look like? How could this simple molecule hold the blueprint for an entire organism? The race for the structure of DNA was on. The race for the double helix. The race had two main fronts. In London, at King's College, two key figures were at work. Maurice Wilkins, a brilliant physicist who had worked on the Manhattan Project, was a pioneer in using a technique called X-ray diffraction to study DNA fibers. And joining his lab was Rosalind Franklin. A formidable, fiercely intelligent and independent scientist, Franklin was a world-class expert in X-ray crystallography. Rosalind Franklin, the woman who saw DNA before the world did. She captures a series of X-ray diffraction images, the most famous being Photo 51, a pattern that clearly reveals DNA's helical structure. But from the very beginning, their relationship was poisoned by a misunderstanding. Wilkins, a shy and reserved man, viewed Franklin as a high-level assistant to his project. Franklin, rightly, believed she had been hired to lead her own independent research on DNA. This clash of personalities and the pervasive sexism of the era created a frosty, unproductive atmosphere. They barely spoke. Despite the difficult environment, Franklin's work was magnificent. She improved the X-ray diffraction technique, discovering that DNA existed in two forms, a drier A form and a wetter B form. And in May 1952, working with her graduate student Raymond Gosling, she took a picture that would change the world. It was a crystal-clear X-ray image of the B form of DNA. She labeled it simply, Photograph 51. To a layperson, it's a fuzzy cross. To a crystallographer, it was a revelation. The distinct X shape screamed helix. The spacing of the spots on the image provided the exact dimensions of that helix. The missing layer line in the middle meant it was a double helix, with the backbones on the outside. Franklin had captured the fundamental secret of DNA's structure. She noted it in her notebooks, but being a cautious scientist, she wanted more data before publishing a definitive model. Meanwhile, 50 miles away in Cambridge, an unlikely pair had formed a friendship. Francis Crick, a brilliant, booming 35-year-old physicist still working on his PhD, and James Watson, a brash, ambitious 23-year-old American biologist. They weren't supposed to be working on DNA. Their lab chief had agreed to leave that problem to the King's Group. But they couldn't help themselves. They were obsessed. Their method wasn't experimentation. It was model building, like putting together a giant molecular puzzle. They knew the basic chemical components. They knew of Chargaff's rules, a discovery that the amount of adenine always equaled thymine and guanine always equaled cytosine. But they didn't know how to put them together. Their first attempt at a model was a disaster, a triple helix with the backbones on the inside, which Rosalind Franklin promptly and correctly dismantled in a visit to their lab. The critical moment came in January 1953. In a move that remains controversial to this day, Maurice Wilkins, frustrated by his lack of progress and his frosty relationship with Franklin, did something decisive. He met with James Watson, and he showed him Photograph 51. Wilkins showed him the picture without Rosalind Franklin's knowledge or permission. For Watson, it was a lightning bolt. He wasn't a crystallographer, but he instantly recognized the telltale helix. He raced back to Cambridge, his mind ablaze. Crick's formal training immediately filled in the gaps. The X told them it was a helix. The other patterns told them the dimensions. Combined with their knowledge of Chargaff's A equals T, C equals G rule, it all fell into place. The two strands ran in opposite directions, with the bases paired neatly in the middle, A with T, C with G, forming the rungs of a twisted ladder. It was simple, it was elegant, and it explained everything. It showed how DNA could copy itself by unzipping and using each strand as a template. As Watson would later write, Francis Crick walked into the local pub and announced, "We have discovered the secret of life." Legacy and Injustice. In April 1953, Nature published a series of 3 articles. The first, a single page, was Watson and Crick's theoretical model of the double helix. Following it were 2 more papers providing the experimental data, one by Wilkins Group and one by Rosalind Franklin and Raymond Gosling. Her crucial photograph 51 was published, but it appeared as supporting evidence for a theory she had been denied a fair chance to discover herself. Her contribution, though pivotal, was cast in a supporting role. Rosalind Franklin left King's College for a new position, where she did brilliant work on the structure of viruses. But her constant work with X-rays, often with inadequate shielding, had taken a terrible toll. In 1956, she was diagnosed with ovarian cancer. She continues to work until her final months, driven by her love for science. She fought the disease for 2 years, and on April 16, 1958, she dies at just 37 years old, never knowing how central her work would be to biology's greatest discovery. 4 years later, in 1962, the Nobel Prize in Physiology or Medicine was awarded for the discovery of the structure of DNA. It was given to James Watson, Francis Crick, and Maurice Wilkins. Rosalind Franklin's name was barely mentioned. The Nobel Prize is not awarded posthumously. Had she lived, it is almost certain she would have shared the prize. Her death sealed her fate as the unsung heroine of DNA. For decades, her story was largely forgotten, until later accounts, including Watson's own book, The Double Helix, inadvertently highlighted her critical role and the injustice she faced. The revolution and the future. The discovery of the double helix was not an end. It was the beginning of a revolution that is still unfolding. Understanding the structure unlocked the ability to read the code. The Human Genome Project, completed in 2003, mapped the entire sequence of human DNA, 3 billion letters of our genetic instruction book. And now, we are learning to write the code. Technologies like CRISPR gene editing are giving us unprecedented power to correct genetic defects, fight diseases like cancer and sickle cell anemia, and engineer life itself. Personalized medicine, tailored to an individual's unique genetic makeup, is no longer science fiction. The story of DNA is the story of every one of them. From Mendel's lonely garden to Miescher's pus-filled bandages. From Franklin's fierce intellect and her ghost in the machine, Photograph 51, to the ambitious model building of Watson and Crick. It is a testament to human curiosity, a cautionary tale of our flaws, and a soaring hymn to our potential. The secret of life, a code written in a language of 4 letters, binds us all. It's a story that began with a quiet monk counting peas, and its next chapter is one that all of humanity will write together. If you loved this journey through science, support our mission. Like, comment, and subscribe for more stories of science.